Good evening, everyone. We're just going to give it a few more minutes before we properly start on account of people are still coming in. Just to make everyone aware, you are all muted with your videos turned off because obviously this is broadcast at large and we don't want any issues with that. So if you do have any technical issues during this, can you please email the events team email address, which is on the screen, and we will use that. We are going to hold a Q&A session at the end of the lecture, which we will take questions for using the Q&A option, which is in the menu bar at the bottom of your screens, or depending on how you're displaying this. That's not the same as the chat function that most of you will be used to from using Zoom the normal way for meetings rather than for webinars. So do use that, and those questions will be moderated, and Dr. McAlister will answer some of them at the end of the lecture, depending on how many we've got. I think we're just going to give it another minute or two before we actually start properly, but just wanted to inform you of all of the settings. And obviously you can use the chat function to talk amongst yourselves during the event, but we will not take any questions with the chat function. We'll only take them through the actual Q&A function itself. Good evening, everyone, again. We just wanted to wait a few more minutes just for the last few people to arrive. And I want to welcome you all to our annual Plymouth Linnean lecture, obviously our first virtual one, which is quite exciting for us to be able to still have one in the current situation. Obviously, last year we had to cancel as it wasn't a viable option to have that many people in one room at the peak of the first wave of the pandemic, and we're thrilled that we can actually continue with one this year. It's fantastic. So if you do have technical issues during this, can you please email the events team email address that's on the screen, and they'll be able to advise you. We will take questions for a Q&A at the end using the Q&A function that's in the Zoom app at the bottom of the screen, which is not the same as the chat function or the raise hand function that you may be used to. So just make sure that you put your questions in the right channel, otherwise they won't be used in the Q&A. And you can talk with each other using the standard chat function, which will be moderated. And we won't take questions from the chat function for the Q&A at the end. They have to come through the Q&A section, unfortunately, otherwise they don't get diverted to the right person. So it's my great pleasure to be able to welcome Dr. Erica McAlister from the Natural History Museum this evening, who I first met a few years ago at an event at the Linnean Society and thought would be fantastic for one of these lectures. And I asked her to come last year, but unfortunately she was far too busy and couldn't do it on that particular date. And this year has very kindly agreed to do it in spite of the fact that she's doing public talks about once a week at the moment, because obviously during the pandemic, lots of people are trying to keep science alive by doing these events. And it's fantastic that she's given us time from her very hectic schedule to do this evening. So thank you very much. And over to Erica for her talk.
Hi, thank you for that. Hello, everyone. Right, let's just share my screen. Okay. Can we all see that okay? Yes. Okay, so my name is Erica McAlister, and well, I would say usually you'd find me at the Natural History Museum in London, but actually even during a pandemic, you wouldn't probably find me at the Natural History in London, because I'm very lucky to work on a group of animals that enables me to travel around the world. I work on the best animals, obviously, because I work on flies. And I realise that this may be an unusual opinion to have with the average person, but believe me, they are some of the most extraordinary beasts you can come across. We have a lot of them in the UK. There's over 7,000 species here alone. And I've become quite obsessed, because obviously the nature of my job, in staring at these beasts. And it's not just the staring at them I like, it's understanding how they function and why they have certain features they do. So this is one of the reasons I wrote the latest book, The Inside Out of Flies, was to talk about their morphology and how it relates to their functionality. Now, being the Linnean lecture, I thought I'd start off with Linnaeus, because he really was the granddaddy of naming. He was the one who managed to sort out the chaos that was before of hundreds of years of us looking at things and describing things in an ad hoc way. And he develops this binomial nomenclature, or he was the one who properly formalized a binomial nomenclature. And he started describing what was around him. And here we go. Here is this description of a housefly, Musca domesticus. And it's quite small. And what we look back on these early descriptions, we go, oh, that pretty much could describe, oh, I don't know, another 10,000 species. So he was good. He started things out, but he really didn't help us out in a lot of ways. Next along was his student, Fabricius, who was a Danish entomologist. And Fabricius really got into insects. Where Linnaeus probably preferred plants, Fabricius took the insects and decided, you know, I can add to the thousands or so that he's done. And he carried on and named just shy of 10,000 species. He really is the father of entomology. But I have a photo here of a robber fly, and this was the auratus, Acilus auratus robber fly. And I don't know how good anyone's Latin is, but again, the description could fit a lot of species. So for us to start looking at species, really understanding these beasts, we needed to develop ways to which we could properly see them. We step back and we find people like Robert Hooke. And now Robert Hooke is an inspiration. He's absolutely amazing. I have a copy of this book beside me because I thumbed through it quite a bit. And Hooke was one of the early advocates of this newfangled technology called the microscope. And in doing so, he was able to properly start looking at all these features that were very important to us taxonomists to help understand which species are which. Now, Hooke wasn't exactly traveling the world, and what he did basically was to use a lot of things that were very close to him. Some of it maybe too close if you've read the book, but he did collect a lot of flies because flies like hang around with humans. We're quite filthy, we're quite dirty, and so the flies are attracted to us. And so he was able to properly start looking at them. This image, this drawing is it's 350 years old, 60 years old, and still you can see the absolute minutiae of detail on this fly's head. So on the left, we've probably got one of the earliest examples of an incorrect taxonomic identification, because he describes it as a drone fly, which is a type of hoverfly, but it's really a horsefly. And we could start seeing the individual facets on the eyes. These are the individual photo units of the fly. And here already you can see that there's a differentiation on the eye with the size of these facets. So it's a really good early example of, hold on, there's a difference in morphology. What's this all about? He went on to, he's also done blue bottles and blowflies and mosquito larvae here. In fact, he describes when he cuts through the larval skin of this mosquito larvae, where he's able to, because they have pellucid bodies, and he's able to look at the organelles within them. I mean, the level of detail is extraordinary. A contemporary across the pond, Swammerdam, in his books are held in Leiden, and I was very, very lucky to be able to thumb through them, and these are my photos of them. He was the first to truly describe what was going on during the life cycle of insects. And again, he picked some very familiar objects, species to us. Here is a mosquito. And what I love about him was when he was using his microscope, when he was making his detailed drawings, he would often correct him. I know quite a few of you have been through the undergraduate process and have taught the undergraduates, and the level of like, oh, I can't do it, I can't draw. And it's like, this was a man with a single lens doing these most amazing detailed drawings from hundreds of years ago. And he corrected himself, he added to it, and this helps us understand the morphology, to understand the taxonomy, to understand the organization, what's going on. And so many hundreds of years later, and slightly less amazing in many ways, because it's just little old me, I wrote about the flies again to try and bring it up to date with all the things that we've really started looking at their morphology. So here is another fly's head. And what we've been very lucky to be able to do now is to image lots of specimens from around the world. So this is a very bonkers Midas fly with a very unusual looking antennae. Now, what we don't know, and what is very interesting, is what a lot of these structures do. We have still so much to learn. So first off, what is a fly? Now, many people get confused with flies because many people go, as a dragonfly, that's not a fly. A mosquito, that is a fly. And so what are they? Well, flies they're one of the most, I believe the third or fourth most diverse group of animals on the planet. There's a lot of them. Okay, and they are part of the insects, and described insects are about 85% maybe of all species on the planet so far to date. I mean, obviously when the fungi people get organized and the nematode people get organized, that figure might dip, but also when the dipterists get organized and start identifying all these cryptic species, it might bloom again. But what makes an insect an insect? You've got 3 pairs of legs and 3 body segments. And then with the order of flies, what makes them a fly is that we work on the adult stage as well, is they have suctorial mouthparts, 1 pair of wings as adults, because the second pair of wings has now been modified to these halters. And these are the balancing organs and are quite exceptional organs, enabling it to fly and do all sorts of crazy stuff. Now, what I love about flies is I've just told you what the blueprint is, and most, quite a lot of flies laugh at that and have completely modified themselves. So you get an absolute range of these amazing creatures. For example, here we go. Here's some rather attractive looking ones. I agree that sometimes my level of attractiveness and yours might differ, but you can't help but fall in love with the middle one. These beautiful hirsute little bodies. It does have your mouth parts, which are not often. And in fact, the mouth parts on the one on the right have been extreme, and we'll talk about those in a bit. So you have quite a range in form, when they're winged, whether they're not winged, etc. But when we talk about flies, what most people think about is just this adult stage. Now, flies, like many of the insects, go through a complete morphological change, a complete metamorphosis from one stage of their life to the other. And in flies, this is particularly important because in the larval stage, there's a lot going on, and this is really useful to us. So here we go. This is a very common, in fact, it's in your gardens right now. Everyone is spotting them. The hoverflies have turned up. Great, spring is here. Finally, we can get in the garden after months of being locked in one room in our houses. And it's really great because these are really important species. But this is an Eristalis. This is a drone hoverfly, and the larvae are amazing because they've got the rat-tailed maggots, and that tail is a really elongate anal spiracle because they breathe out of their bottoms. Now, this is really useful because they often live in really dank, stagnant environments, which is quite anoxic. And to enable them to survive in these environments, they can stick their little tube way up to the surface and able to breathe from the open air. And they are massively good. They're incredibly good at getting rid of our decomposing waste. So the larval stage is actively getting rid of this, where the adults, especially in this case, are really important pollinators. In fact, one of these species, Eristalis tenax, is arguably in the top 10 pollinators in the UK. Flies have always been neglected with all of their roles, but we're beginning to understand more of them. So this amazing morphology is enabling them to live in weird places. Who doesn't like their morphology? Again, here's some more common larvae in your garden. Oh yes, look at those cute little faces. But no, be careful. Those once more are your anal spiracles. Yes, you are looking at the backside of a crane fly larvae here. And these are quite extraordinary beasts. And we use these features to identify them. So as you can see, they're quite distinctive, and some of them are quite amusing, and it enables us to look. And this is great. We complicate things by breathing and eating from the same mouth, from the same orifice. Why do that? Why don't you separate this so you can feed with one end, breathe with others, and then you can get a job done much more efficiently? And yes, they do things efficiently. These maggot frenzies, which everyone thinks, oh, it's revolting and disgusting, are actually really useful. And they are one of the things that we use for, say, forensic science, when maggots first arrive on a corpse, these sorts of things. But we also use them for decompositional studies, and we use them in medicine. So the fact that this stage is like this is because they are the feeding stage. This is a very important thing in nutrient recycling, etc. And they are very simple. Their form is very simple. They don't need legs at this point because most of the time they've been dumped exactly where the food is, be it on the body, in the body, around a compost, in a tree hole, you name it, these little maggots can get it. But it is the adult stage that everyone is more familiar with. And these are very complicated stages. These are very complicated morphology now, where you've got the head, thorax, and abdomen, which is much more defined. Now, the head part is where you've got most of the senses. So this is where they have their visual senses. This is where you've got their hearing, you've got taste, you've got all these things. But don't just think it's always located on the head, because that's not true. In fact, flies can taste with their wings, they can taste with their feet, they can even taste with their naughty bits. All sorts of things going on because they have senses everywhere. But here is a lovely example of two big-headed flies. And these, obviously, their senses predominantly, I think you can figure out, is visual. And what is really lovely here is that you can actually see that the lower one, which is the female, they're in the process of copulation. Sorry, with flies, you're never far away from copulation. She's got enlarged facets at the front of hers because those enable her to find her prey. Or not, right, I should really correct myself, not her prey, her baby's prey, because her larvae are parasitoids and she needs to actively find the host to let eggs in for them to develop. So usually the men have the big eyes, okay, because the males, they're the ones who have to go and find it. And the females, they just sit back and wait for the male to appear. But when the females are active hunters or active collectors of prey for their larvae, then they will get this development. Here you go. Don't always think that the best predators on the planet are the biggest one. Every time there's a documentary about predators, I get really angry. This is a finger, just to give you a scale. This is a little robber fly. These are some of the most aggressive, amazing predators on the planet. And I'll get onto them in a minute. But eyes aren't just used for seeing, they're used for flirting. A fly is never far away from a flirt. And here we've got a lovely example of stalk-eyed flies. And the males would do what deer do. They will rut, they will defend territories, and they will show off for the ladies. Now, mouth parts is also associated with the head. Not all of them have mouth parts. This little cutie doesn't have any mouth parts as an adult because it's the larval stage that does all the feeding. It doesn't need to do it as an adult because the larval stage, in these examples, these are bot flies, and the larvae either live under our skin, in the stomachs of weird creatures, or in the nostrils. This one is a sheep bot fly, and it's found in the nostril of sheep, obviously, and entertaining. In the UK, humans can get it as well. Lots of cases in Wales, unsurprisingly. But what's going on is that this is a really nice scanning electron microscope work looking at, CT tomography, looking at what's going on inside the pupil, larval pupil and adult stage of this fly to show you what's going on with its gut. Because the larval stage, the gut needs to be huge because it's eating so much. And this basically disintegrates during the pupil, during the complete reorganization of the body tissue. So when the adult, the newly emerged adult comes out, it's vestigial. It's basically not even there. And some of these, because they live in such weird habitats, may be really critically endangered. This is the rhino bot fly. This lives in the stomach lining of a rhinoceros. Arguably, therefore, it could be one of the most endangered species on the planet. But most of the time, they do have mouth parts, and some of them have exceptionally long mouth parts. This is a mouth part of an emestrinid, a tangled vein fly. And if it was a human, your tongue would be 6 meters long. Now, what is extraordinary about these, unlike butterflies or moths, is that they cannot roll them up. They cannot curl them up. They, in fact, articulate them under their bodies. So they will fly along with their heads down with this enormous mouth part coming out. Here's the UK's own version, these little bee flies. Oh, I've got to say I love these. And these are appearing all over the place now, which is great. And these have co-evolved along with long-tubed plants to enable them to get to the nectaries at the base of the plant. So they have some beautiful long mouth parts to enable them to get into the parts of plants that other species can't get in. Bees are not always the best pollinators because a lot of bees, the honeybees, are short-tongued, so they cannot reach deep down into these nectaries. But the mouth parts that you will be more intimately associated with are the mosquitoes. And these are incredible. And I know you don't like them, but they're turning out to be very, very useful. Because actually the ability of a mosquito to penetrate your skin actually takes some blood from you and fly off to enable her eggs to develop, we are actually using for smart needles now. Because she's able to penetrate your skin without doing so, they've been looking at this. And what they've discovered is that she's basically a pneumatic drill. And in piercing your skin, in vibrating onto your skin, she's able to penetrate with a lot less force. I believe it's about 400 times less force. So we are now making smart needles to enable us to use this technology for us. Think about those mass vaccination programs right now. And if we suddenly rolled out all these amazing needles that didn't need to hurt us so. We have the antennae. The antennae are the other major sensory part on the head, and this is where they hear. So they hear with these amazing organs. We don't quite know what's going on with a lot of them. Some of them are really quite ornate, but we do know that they're very, very important, and some flies have incredible hearing. One of them, though, the antennae are not on the head, they're actually on its shoulder pads, as it were. And this is a tachinid. These are parasitic flies. And these, again, the female has to find a really active host for her larvae. In this case, it's a grasshopper. And now you've tried to catch a grasshopper. Obviously, all of us have, haven't we? And you know how difficult they are. And so what she has to do is listen. She's like very, very small. So how on earth can she listen and triangulate exactly where this grasshopper is? Well, what she has in her shoulder pads is she has this little rocking mechanism, and this rocking mechanism is able to amplify where the noises come from and enable her to triangulate exactly where it is. Now not me, because I'm not that clever people have looked at this and then gone on and developed very tiny loudspeakers based on this system, and also are beginning to develop very tiny hearing aids to enable us to be able to hear with such acuity. So we're onto the thorax now, and this is generally where the locomotory part. So you've got legs, you've got wings, you've got everything to enable the fly to move. But it's also really bristly. Bristles everywhere. And bristles are really important to us for identification. Yep, I have to count all those bristles. I have to see which way they're going. I have to count the length, the colour, you name it, I have to do it. Bristles drive me mad. But bristles are really quite important for the fly. We're only beginning to understand the true functionality of them, but there is a very clever fly. And it's a scuba diving fly. Now, this is lovely. We get a lot of scuba diving insects. Yep, there's water beetles. They scuba dive all the time. They do exactly what this does. They create a plastron, an air bubble around it. And this is created by, again, the chemicals that are exuded from the exoskeleton, as well as the hairs. But this one is extraordinary because this lives in the Mono Lakes of California, which is the most saline aquatic environment out. It is incredible. It is so concentrated that if all these other insects went in, basically it would completely disrupt their membrane and create disrupt the water bubbles. So they wouldn't be able to breathe underwater. This fly, physicists have worked out that it hits the water at a certain angle, 45 degrees exactly, and she's able to then now create this plastron, this water bubble, air bubble around her to enable her to get to the bottom where she can lay her larvae, which quite happy live in there. Now, this creates an environment for the larvae that is unique in basically there's no predators, because nothing else can really live there. So you've got all these dead animals that have, come to the surface or hit the surface and are floated down and gradually decomposing, which is creating a massive smorgasbord for these little larvae. What I really like as well, have a look at this photo. The eyes aren't covered up. So she has basically the opposite of swimming goggles to enable her to look around her in the water. The legs aren't just used for walking. Oh no, they're used to flirt. Look at these hairy legs. We're not quite sure what's going on, but obviously the females love it. Don't love hairy legs as much as we love a good gift. And gift giving is a very common thing with flies. There's a whole family, in fact, several families that do love giving gifts. And this is Empids, the one on the right. This is one of the males. And you see he's got expanded feet, and because those are where his silk glands are. Because he actually creates little silk bubbles where he wraps a dead fly to provide her for a little gift, an amuse-bouche, before they proceed with their copulation. It's very romantic. But flying is what flies are known for. Flies, it's in their name. You see what we did there. And so they've gone through all this amazing period beforehand to enable them to disperse and reproduce. This is the crux of what they're doing. And lots of them are really, really good at flying. In fact, these are very, very famous flyers. We're now beginning to look at how much these hoverflies are actually flying around. There are species that migrate across from North Africa. In fact, they're turning up right now. The marmalade hoverfly, the Epicephalus bultiatus, which is annoyingly cut off on my screen in the top right. These are turning up in your garden. Some of these will be migrants from around the world, from North Africa and Europe, and some will be our own homegrown ones. And they're really important for plant genetics. They are spreading the pollen around. They're doing all sorts of amazing stuff. And they're really good. We're looking at how they fly, how they hover, and we're using this to develop increased technology for our own aeronautics. So I know a guy in Imperial who's looking at jump jet technology and supercomputers based on how hoverflies fly. But they don't all fly. No, because that would be far too easy. Some just walk. They're not called walks. And this is one of my favorites. These are absolutely adorable little teddy bears. And these basically are, they've got no antennae. Oh, they're slightly blind. They've got no wings. They've got no halters. And these are found on the back of a bee. In fact, this very specimen is sitting on my desk right here. And these are quite extraordinary. And they are only found on honeybees. And predominantly they're found on the queen bee. And they are kleptoparasites. So they don't actually steal food. Like, they don't actually cause any real detriment to the bee. They go round to her mouth because when she's being fed by all the workers, and they sneak bits of food there. Just think of it like if you've got a beard, it's like a little beard cleaner. That is what's going on there. And these, sadly, are one of the few side effects of adding the antimiticides, or the miticides, sorry, into beehives. So we didn't get rid of the Varroa mite, but we definitely got rid of this. So luckily, in the last couple of years, there's been a few instances where these are turning up in hives now. So it's quite exciting. But we've only just discovered really what's going on because, you know, in proportion to body size, they're quite large in comparison to the bee. So why on earth is she letting maybe 1, 2, 10 of these little creatures? Can you imagine little toddlers running around your body, holding on for dear life? Why is she doing that? Well, actually, these have produced chemicals that are absolutely identical to her own chemicals. Okay, so they've managed to mimic this. Now, this is amazing. We're beginning to look at this because this can have huge impacts when it comes to biological control. If we can persuade insects that they do want to come or don't want to come to certain areas. I have an inordinate soft spot for parasites. These are flightless flies. And I collected the ones on the left from Dominica off bats, which you can see from the image on the right. To be fair, that's not the biggest bat in the world, and it is a fairly large fly, but it's cool. And why these are amazing is because not only are they flightless, they've therefore lost all the muscles in their thorax associated with flight, but they have massive claws that they hold on. And when they are not feeding, because there's no back muscles, their head pings out the way. Extraordinary adaptation to their host and the environment they're in. What is very clever is these flies give birth to live young. We never think of anything apart from mammals really giving birth to live young, but these ones do. In fact, on average, these group of flies will only give birth to about 4 or 5 flies in their lifetime. They really are nurturing mothers. She has internal lactating glands that she will use to feed and nurture her larvae. And it's only at the final bit of the larval development, when it's just about to pupate, does she leave her host, find a wall or the cave or whatever, and she lays her egg then. So she is quite a caregiver really when it comes to it. And it's quite unusual. This milk, this lactation that she's providing is actually quite similar. They've been talking about milking cockroaches. I don't think we're going to be milking cockroaches or these bat flies anyway soon, however. So we move on to the final part, the bottom, the abdomen. This often isn't as attractive as the head or the thorax. I have to differ. These are a group of flies called Stratiomyids, soldier flies. These I affectionately call the fat bottom flies. And I think this one is a beautiful example of what. And often this is the stage which gets a bit funky though, because of what goes on. So this is all more about the internal bits. So they have to advertise, the males and the females, to each other about their prowess. She has to advertise that she can actually hold a lot of eggs and bring them full to term. And he has to advertise that he's worthy of being the father of her offspring. So most of the time, when it comes to taxonomy, we are basically fiddling with genitalia. And this is because this is the part that changes so fast in comparison to all the other features. So this is the structure that we're comparing. We know we've got a new species if its genitalia is different. So Linnean started it. We started with males, always describing the male as the type specimen. So every time there's a new species to science, you would have one specimen that was that name-bearing specimen. And more often than not, it was a male. I've been around so many dipterists for too long in my life to, finally get over being told that female genitalia is really dull. And I genuinely disagree with this. There's quite a few families of flies where female genitalia is really quite cool. These are for example, the females that have massive long ovipositors. So see this massive hook here? This is the ovipositor. This is an egg-laying tube, because she actually has to penetrate into fruits or things like that, which will be quite thick, and she has to be able to insert and actually make sure her egg is in the right place. And at the end of those ovipositors, she's got senses. She can actually sniff out which is the best area to do. Amazing stuff. My favorite genitalia for females is one that's used for slicing through the backs of ants, but I thought that would be a bit grim for a Wednesday night. But of course, we must talk about male genitalia, because it is quite extraordinary, and flies seem to have run away with having some of the most outrageous aediagus, what we actually call the penis. So here you go. There's some amazing corkscrew designs going on here. These remind me of those machines that buzz when you go too tightly for them. And for many years, we were a bit confused about what's going on. I love the one in the top right. My colleague in America sent me this going, oh, you think they're weird, Erica? What about this corkscrew? It does leave me thinking. But this is quite extraordinary, because we're beginning to see what's inside the females and realize actually she's way more complicated than we thought. There's this huge competition going on between the male and the female. There really is a war between the sexes. And all of this has led to some extreme adaptations. But it's not over yet. So once they've managed to copulate, once she has become fertilized and he's done his bit and he's gone off, there's some still stranger things that have happened. Here we go, arguably one of the weirdest looking flies you're ever going to see. Yep, absolutely. It's like a pear with a base. And what's happened here is that the female's copulated. It's a bat fly, by the way, but she starts off relatively normal. She's got wings, she's got, legs, she's got a head. I mean, everything that kind of makes you look like a fly. She's copulated, all's good, found a host, and then gone okay. Now, her mouth part is quite chunky anyway, and she shoves it into the side of the bat. All good. Starts feeding, then rips her legs off, rips her wings off. So she kind of looks a little bit more slug-like now. Oh, no, she hasn't finished there, has she? No, she invaginates her abdomen around her thorax and her head. So that's what you're seeing here. This weird pear is actually her abdomen wrapped around her head, and that little smiley face, yay, you've guessed it. Once again, it's her abdominal spiracles. It's amazing. Why is all this important? Well, we need to make sure we're looking at things properly to get their names right, to start understanding how their ecology fits in, how they fit in the world. Flies are always getting badly labelled, okay? Everyone blames them for every single crime in the environment. They're dirty, they're filthy, they're massive vectors, and yes, some of them are, but a lot of them are really important. So here we go. Here's many examples of everyone getting them wrong. Everyone saying, oh, look, aren't they beautiful bees? What amuses us more is this bees of the world. And the publisher has put a fly, which isn't even it's not even a hoverfly. This is a housefly on the front cover. And so everyone's like, oh, the bees are so important. I'm like, no, they're not. In fact, we've got to pay more attention, because
in her 2017 work, The Secret Life of Flies, The entomologist Erica McAlister estimated that for each person on Earth, there are how many flies? The figure in question is about the population of Zambia or the Netherlands. You can have 3 million either way.
6 million?
No, it's 17 million. Amazing, isn't it?
Yes, it is amazing. Because these many different flies are out there doing many different things, and they are really important for so many different reasons. So I'm just going to finish on this last little story about an acyllid. This is a robber fly, and this is a UK species of robber fly. I only got to see this for the first time a couple of years back, and I definitely squealed. Now, I talked about robber flies right at the beginning. They've got amazing vision, absolutely amazing vision. But these are also exceptional flyers. Okay, they're able to yawl, they can turn, they can balk, they can do all sorts of things. And we've been studying them because they're so fast at being able to react and how they can predate in such a way. But what is amazing about them is a further adaptation of them. These are venomous flies. Okay, these are part of the neglected venomics group. They have venoms that are new to science. We've been studying the bees and wasps and spiders and all of these creatures for a long time, but we're suddenly realizing that actually a lot of the predaceous flies too must be venomous because they're able to subdue them. In fact, in America, there's like one of these flies that's able to basically destroy an entire hive of bees by itself, and it's also been seen to actually kill a hummingbird. So being able to predate, attack, subdue, and predate on a hummingbird is a truly unique thing. So if you don't remember anything else for tonight, I'm going to teach you some venom names. This family is called Acylidae, and these new venoms to science, there's at least 10 of them, we've named Acylin after the fly. So you now know Acylin 1, Acylin 2, Acylin 3, and so on. So there's so much more we can learn about these flies, so much more we can study about them. So I just want to say thank you. Thanks to all the flies that have been an inspiration in my life, and thanks to everyone who's been carrying out all the examinations, and may they continue. Thank you.
Okay, thank you very much, Erica, for a wonderful talk. It was very entertaining and quite informative. We've got 5 questions so far queued up in the Q&A system. So if you've got further ones that you wish to ask, please type them in so that we can answer them. In the meantime, while people are just doing that, we have 3 copies of Erica's latest book to give away, which she's kindly going to personalize and sign and send off to you. But because of the lockdown situation at the moment, that might take a little while until we can get back into workplaces and so on to actually do it. So during the lecture, I have gone through the list of attendees, scrolled through it at random and picked out 3 names totally at random. And obviously we've got your contact details from your registration. And those 3 signed copies will be going to Bronwyn Ferguson, Jennifer Poole and Dr. Nigel Marley. And we will contact you tomorrow to arrange all of that so that we can start that off. So in terms of questions that we have had, let me just move my light switch so I can actually see my monitor. Bear with me a second. First one that we had, which is from Mark Papp. You mentioned antennae as sound receptors. Are they chemoreceptors as well as sound receptors?
Yeah, we think they are. It's not really my specialist area, but yes, they are. We know that they've got in their sensilla, they've got chemical sensilla as well, so they can taste with them.
Okay, we've got several different ones from Sarah Collins, so I'll interlace them with questions from other people. First one is, do the bats try and remove their passenger flies, and where do the offspring emerge from the flies? Is it on the bats?
No, so I don't know how the bats handle them, because believe me, when you try and catch bats to try and catch these flies, they're more angry with you than they are angry with the flies on them. And I don't know if you've ever seen bat look disgusted at you, but that's the usual look they do for me. So they look like, when they're on the bat, they look like drunken spiders, the way they're kind of crawling around the bat, and they're like, ah. And most I've seen 2 or 3 on one bat, but I believe you can get loads of quite more. Now, with these ones, the larvae aren't on the bats. So the female will actually reverse off the bat. So when the bat is dangling down in its roost, be it a cave, be it a wall, a tree, etc., she will crawl off onto that substrate, and the larvae is just about to pupate. So she basically gives birth, it hardens, and then she reverses back onto it to make sure she wedges it completely on the side, and then she herself walks back onto the bat, and she will hopefully, if all things go for her, stay on the same bat most of her life, or stay on a bat most of her life. And when the adults hatch, when they've pupated, they will have to quickly scramble to find their own bats.
Okay. Next question is from Andrew Sorosi. In the last fly that you talked about that was venomous, what's the nature of the venom that it uses?
We believe it's a neurotoxic one. It doesn't dissolve. It's not like a necrotic one, but it paralyzes. There's some really horrific descriptions of early experiments where they start using it, and how they get all the other insects to watch what goes on, and they use a lot of bees, and it's just if you ever want to, just look up venoms in flies, and you can read some of these. So it paralyzes them, it enables them. So they're not like Hymenoptera in the same way that they will paralyze their prey and leave their larvae in that. When the flies are parasitoids, a lot of the time they let their host carry on as normal and just eat its insides out, which is nice.
Flies are nothing if not glamorous.
I love them.
Next question from Dr. Nigel Marley, who won 1 copy of your book. Are any flies parthenogenetic?
Parthenogenetic?
Yeah, I thought you'd say that.
What's that?
I'm going to Google it because I don't actually know myself, to be really honest with you.
Parthenogenetic?
It's not a term either.
Oh, parthenogenec.
Yes,
that's right.
Sorry.
Okay, parthenogenec. How are they? I think there are. I think it's in the cecitimids, which are the gall midges. Now, this is really taking my brain and making it work. They are, I believe it's in these that we do get some. So in some of the more, I don't like using the word primitive flies, but the ones that appeared on the planet earlier, I think we've got some examples of that. Not many, really not many. Obviously, the aphids have taken that. And what we mean by parthenogenic is you're producing clones of yourself. So the aphids have taken that reproducing strategy and run with it.
Okay, next question I have is from P. Kennedy, which is on invasive alien species, which can be extremely disruptive to insect communities. And some of these invasive generalist predators, or some of these are invasive generalist predators, where diet analysis suggests that the flies can form a major part of their diet. Would you anticipate, therefore, that fly populations will ever be significantly impacted by invasive generalist predators, or does their reproductive potential always enable recovery?
No, I'll go with the former, not the latter. We've got so many specialists in flies that have niche habitats, and I found in a few pockets of area that if you've got an invasive that was basically an oligotrophic, they ate everything, they are just as likely to disappear than any of the other insects. So it will be very, very detrimental to a lot of them.
Okay, next one, another one from Sarah Collins. When did flies first emerge in the fossil record?
Around 260 million years ago, somewhere around that. Obviously, we haven't got the most primitive flies. We're still actually arguing which are the basal flies, which is great. So it used to be, when I was littler, it used to be crane flies were the most basal flies, and now they're like, no, crane flies aren't basal anymore. They're quite quite advanced, and it's like, oh. And so in trying to establish which are the most primitive flies is quite difficult. But we're going on about 260 million years at the moment.
I can sympathise as someone who works on some of the oldest bacteria. What the oldest bacteria are has changed about 10 times a year.
Oh, well, I love it. Insects aren't even what we thought were insects now.
Yes,
and it's just like, oh, we're a crustacea. I've got to stop saying we, because obviously I'm a mammal. But it still changes, and that's the whole thing about this discipline. It's not static. It's really dynamic.
Okay, I've got another question. This one is from Lewis. This is on a topic that we've talked about in the last few days. Is it true maggots can clean wounds by consuming necrotic flesh?
Yes, and in fact, you're very lucky. On the NHS, you can have this done. It's really cool because as I've got a maggot here, these are giant microbes. You've seen all giant microbes. You must have giant microbes, Rich. Right, so I've got my own ones. And what we do now is, Genghis Khan travelled across Asia with a wagon load of maggots because he knew about the debridement ability. And so certain species that are able to just eat the necrotic flesh to reduce gangrene and all the infectious pathogens, bad bacteria that are getting into your wounds. Now, we're realizing now that this is actually really, really important, especially as more people are becoming, as penicillin is no longer effective in the same way that it was. And MRSA and all these things are problematic, and type 2 diabetes, interestingly, because that creates massive ulcerating wounds that are very difficult to heal. Bingo, the maggots. So the maggots are able to come in. The maggots, nowadays, you don't have them running around your flesh because apparently people are a little bit sensitive. They come in what looks like a tea bag, and only their little mouth parts at the beginning, these two tiny little mouth parts, they've got a really reduced head at this stage. They're the bits that poke through the tea bag and they actually munch away at this necrotic decomposing flesh. At the same time, they're able to release enzymes that actually not only get rid of this flesh better, it stimulates the growth of your skin. So after this, go away and have a look at YouTube videos of when you've got maggot debridement therapy, and you can see that the wound is cleared it's healed much nicer. So absolutely recommend it. Cardiff, there's a nurse there that's the champion maggot nurse, and she's trying to get all the other nurses to get more involved with this maggot debridement therapy. It's really, really good.
It is, and it's absolutely amazing results. It's been around in sort of modern medicine for about 30 years, but uptake hasn't ever been very good because flies.
It's been around a long time. American Civil War really launched it because there was a lot of bodies hanging around, as it were, and a lot of doctors on site. So war is really good for medicine. Funny that, terrible for many things, but they were able to study a lot of what was going on, and they realized that all the soldiers that had flies in their wounds actually healed better.
Okay, I'm going to combine sort of two questions and ask them at the same time, one from Paul Hardy, one from Janet Vosper. So part one is, with global warming, are we likely to see malaria in southern England? And part two, are flies reducing in numbers along with other insects?
Okay. Yes. So we have 5 species of Anopheles in the UK. So there's about 34 species of mosquito here. Now, we did have malaria in the UK recently, in fact, during the wars. And it does turn up quite regularly, thanks to, you can guess why it turns up quite regularly, around Heathrow and Gatwick. Wonder what's going on there. Yes, so we have these species that will be able to do it. Actually, malaria isn't the one that concerns us mostly, because we have anti-malarials that are quite effective. It's things like dengue. Now, dengue itself, the mosquitoes, there's a good population in the southern front, and dengue has now turned up there and is surviving in those populations. And southern France really isn't that far away. But you'd be glad to know there's a UK mosquito surveillance organisation. It's just the best superheroes, really, isn't it? And they go around basically checking out all the mosquitoes everywhere. And they have surveillance system all around the UK. They have a monitoring system. So if any bad mosquito or a vector that is potentially able to carry something we don't want turns up in the UK, they can go and sort it out. Second thing, global warming, what will happen to lots of the flies in this case? Well, we don't really know, okay, because everyone has just been studying all the other creatures, which are the charismatic megafauna or the charismatic mini fauna. So butterflies and ground beetles, which are good, but actually they can't really tell us anything because they do not exist in loads of different niches. Flies get everywhere and do anything. So what's going to happen with flies, we're not totally sure. Some cases they will actually absolutely they'll be destroyed, but that's because of their land is going. So land use change, things like that. But some of them are doing better. Now, that hoverfly I talked about that came from North Africa is actually benefiting at the moment and it is bulking the trend because because it flies so high, it flies in the aerial plankton, and it is able to see pockets of dispersed plant populations, so it's able to find food sources where a lot of the other insects can't. So it itself is actually being a conservation hero as well by spreading plant genetics. So there's some of them that will do better, some that won't do.
Okay, next question is from Salvatore Giordano, which is, how significant is the contribution of pollinator flies versus pollinator bees, and could they and are they be used in agriculture to the same degree as bees and bumblebees?
Hey, flies are amazing, mate. You have a cup of tea, you got to thank the flies. If you have chocolate, you've got to thank the flies. Mangoes, leeks, carrots, so much of our crops are actually really dependent on flies. We have about, I can't remember the actual figure, but it's about 550 interactions we know are definitely going on, but there's a lot more to go. Of the fly families, at least half of them have got pollen feeders. So they're probably pollinators. We know that they're incremented by feeding on pollen. So really, when it comes to it, now what they're different to the bees is that they don't have to go back to their nests. Flies just are selfish, don't care about the others. They're like, yay. So in terms of they may not be able to take as much pollen in one feeding event, they have many more feeding events than a bee that has to go back. So actually, when we start looking at it, flies will become very important. Flies don't worry about the cold. Your little bees are like, ooh, I'm too cold, can't do with it. Flies are like, okay. So in alpine environments and things like that, we already know flies are some of the dominant pollinators. You've got to think about nocturnal pollinators. Lots of flies fly at night, all of these different things. We are really just beginning to skim the surface of how important flies are as pollinators.
Okay, I'm going to combine two questions again, one from Dr. Sarah Collins and one from another Sarah. How many flies are parasitic in terms of sucking blood as a rough proportion? And are there any flies that are extremophiles?
Yes, to the extremophiles. There's flies that can actually be frozen, which is quite cool. In fact, there's a fly that can go to sleep for 13 years. It's a chronomid, and they're looking at it, how it does that, how it's able to desiccate itself and things like that for space travel. So we've been sending flies into space since 1947 because they're so weird and amazing and adapting. So we're beginning to look at these. What was the first part of the question? Oh, how many are sanguivores, bloodsuckers? Right. So in terms of species proportion, I don't know, 10%. So it's, apart from one family, it's only the females that do it because they need it for the egg development. Tsetse, you get males doing it as well. So the males and females. So in the higher flies, the Tsetse do it. You get the horse flies, the females do it. Mosquitoes, obviously midges. You get a few basal lineages of some of the other flies that have gone on to be predators that are like a bit of blood, but that's not it. I mean, it's basically when why they do it is because blood is the best food source. So it is definitely a way for doing it. But remember that she's only doing that for egg development. She doesn't use the blood herself. She actually needs pollen. She needs nectar. So they're important pollinators at the same time.
And you'll be impressed with my segueing skills now. Related question from Billy Pike. Why have horsefly larvae evolved to be such effective predators? Is direct larval predatory behavior a common fly tactic?
Quite a lot of them are. Again, as a food source, another animal is a nice food source. So horsefly larvae are venomous as well. So they're really good at subduing their prey. There's beautiful anecdote of a BBC reporter in America feeding frogs to horsefly larvae, which obviously you're not to do. That's totally inappropriate. But these massive frogs, not even like tadpoles, massive frogs that they're feeding, and the little larvae is just like properly attacking it and taking it down. The larval stage in the flies is the predominantly feeding stage. So this is really where they've got to lay their fat reserves down. This is where they're creating the energy for the adult stage. And an adult horsefly is an extreme flyer, and they can cover a long distance. So these larvae, it pays for them to be highly successful predators.
I have a lot of sympathy for horseflies. I'm one of very few people who's quite happy for them to feed on me because I don't think it's really that bad. And they like it. At the end of the day, they need blood to survive, and I don't really care if it's mine. Question from Grace Coveney on the so-called scuba diving fly larvae. How much of their life do they spend in the water, and how is it that they've kind of learnt to dive at such an angle that they can form an air bubble?
To the latter part, I've no idea. This is a part of evolution. I'd love to see the thousands and millions of failures before actually something successfully happened. Larvae were able to survive, and this trait became embodied within the species. Who knows? I'm sorry, I am not studying those. What was the question about the larvae?
Let me just go to it as I've moved it to the other category.
How do the larvae survive?
Won't be a second. Scroll down to it. It is how much of their life?
In this case, the majority of their life. In fact, with most flies, the majority of their life is the larval stage. They talk about longevity of flies. The one I talked about, the chronomid, it could spend 13 years. But like 12 and a half of that, 12 and 3/4 of that is the larval stage. So it is this, especially when they live in quite extreme environments, they can have really prolonged larval stages. And many of that can be periods of inactivity. So they could just go to sleep. The same with stag beetles in the UK, which the larval takes like 5 years because it eats rotten wood. You get a lot of flies that have exactly the same habitat. And wood is just so nutritiously poor. I mean, it's great if we have some nice microorganisms, which is probably the only nutrition they're getting from it. But there's some beautiful timber flies, which are massive flies that don't feed as adults, but their larvae spend 5 years doing exactly the same thing, trying to get it. So again, with these saline flies, not that long at all, but more of its life than the adult.
Okay. I'm going to take we've got 4 questions queued up, and I'm going to just take just those 4 because otherwise we'll be here forever. But obviously people seem to really enjoy this. We're getting a lot of comments coming in that are very happy punters, which is really nice to see. Thank you all very much for that. So last 4 questions that we got. One from an anonymous attendee who first of all says, what a fascinating lecture, thank you. And they heard on Radio 4 programme that when flies lay eggs on cat food, cats will not eat it because they can detect something that the eggs emit. And they're wondering, in rural environments, are there other animals that can detect these chemicals emitted by fly eggs and avoid eating the food that's been
we presume so. These chemicals, are actually what we're thinking about using for biological control. So remember that female with the massive ovipositor? She will actually leave a chemical signal behind to say either this is a good habitat for your larvae, come and do it because she wants strength in numbers, or she would say, don't bother, this is rotten. So they're leaving little signatures all over the place. In fact, flies leave signatures everywhere. When a fly lands on the window, it's leaving signatures behind. Those flies on your lampshade, it's because there's signatures left behind from other flies. And the other flies are curious to figure out what flies have gone on and told them before that. So all of these little things, there's chemical signatures. We're only just beginning. All these chemists need to get themselves into gear and start figuring out what they're all saying to each other.
And so the last 3 that we've got is one kind of similar to one we've already had, which is regarding the sort of relative proportions of pollinator bees versus pollinator flies. And is there an estimate in terms of what percentage of world pollination is fly rather than bee?
No, good luck. If you give me a lot of money and a lot of time, then I can tell you. There you go. We have no idea yet.
And the last two, one from Matthew Wharf. Does there seem to be any benefit to the rigid proboscis of bee flies rather than the rolled mouth parts in butterflies?
They're not totally rigid. They can bend and they can be flexible. So they're, you know, I'm not sure they, in fact, a mosquito mouth part, that long proboscis, that bends under your skin because it sniffs out where your capillaries are, which is quite a thought when you're drinking, when you're having your blood sucked from you. But they are rigid. I presume it's because of strength. I presume it enables better capillary action in some ways. Again, you'd have to probably talk to a biophysicist to understand more about the capillary action of mouth parts.
And the final question, which is one that I had myself as well, how many flies have actually been genome sequenced?
Very few. Arguably, there's probably more flies than most other animals have been sequenced. The first genome was sequenced from a fly. Okay, and we were quite happy with that. Obviously, the model organism for all animals is a fly, Drosophila melanogaster. So this has been 100 years helping us out, and we got the first genome from it. And arguably, a lot of the fly species are really important vectors. So your mosquitoes, there's a lot of genome sequencing going on there. Although we've got a long, long way to go. The museum actually is involved now in a UK genome project of trying to sequence, theoretically, most of the biota of our country. So that's a lot of species that we want to get genomes for, which would be incredible if we even begin to get like 10% of that.
That would be amazing. Coming from the world I do, which is bacteria and archaea, we've probably got about 60% of known species sequenced now in the bacteria.
Wow, really? 60%?
Far fewer in the archaea, but in the bacteria, probably 60%. But that's only the type strains of each species. And we have hundreds of strains of each species. So we still don't know much about diversity within species.
Yeah.
We're finally sorting out like what the families and the orders really should be, using genetic evidence now and not color and colony morphology and things like that, which is what we used historically, and it's sorted out a lot of mess. But we're having to use the whole genome for the bioinformatics, which is great for us because we're dealing with genomes of between 1.5 and 15 million base pairs. But when you're looking at even a small fly genome, it's astronomical in size.
Some of them aren't. Some of them are really quite small, which is quite amazing. It's like, how are you managing that? And it's like, I'm a fly. I'm quite clever.
Still got all the stuff like introns and all the intergenic spaces that we don't have to deal with. So
yeah,
I always think bioinformatics in the eukarya is so much harder just at base level to start doing anything because you've got so much more to deal with and the cost is so much higher. I mean, we're paying 60 quid a genome these days. Yeah,
which is amazing considering what it was.
Someone's asked, why is there a halo around my head? That's the light fitting in my I do not qualify for a halo, believe me. So thank you all very much. That is our last question answered. I think we had about 19 questions, which is pretty good going. We've managed to answer. I don't think we've ever had that many in a live lecture because people don't like putting their hand up. So it's kind of nice doing these things virtually because we get that. And we had lots and lots of comments from people as they're sort of starting to log off, thanking you and saying what a fantastic lecture. Someone suggested we need to have a TV series on flies because they're so underappreciated.
Yes.
And people are all saying they've learned a huge amount about flies and they're very happy, which is fantastic. So thank you all very much as an audience for coming and for entering this brave new world of webinar versions of our public lectures, which is fantastic that we can do them. And on behalf of the school and the university, thank you very much, Erica, for giving the lecture.
Thank you very much for having me. It's a pleasure.
You're welcome.